So this other work I started during my PhD in University of São Paulo. And now as a postdoc here in Texas A&M, I expanded the application to polyploid species. And we have this idea when we start to work with sequence-based markers, because they have some characteristics like they are many, and they come from different libraries. What is good, because we can adapt the protocols according with our needs and our species. And maybe the most important characteristic is that it has lower price compared to other ones, but lower the price, higher the genotyping errors that we can find. And this can be a problem, as the previous speakers also mentioned. And working with these types of markers, we also require more bioinformatics skills, because it doesn't come ready, and also computational resources to run the analysis. And because of the genotyping errors, what we see is this wrong number of recombination events. So this is simulated data. And here in the bottom, I simulate a mapping population. So I have the two individuals haplotypes, the progeny haplotypes, and just as example. And in the left side, I could, with the mapping software, I could reproduce exactly how I simulated and have a nice map and a nice recombination fraction matrix. But in the presence of genotyping errors, the mapping software cannot reproduce the haplotype of these individuals. And I have a messy linkage map, and the recombination fraction matrix clearly presents some wrong thing. And because doesn't make sense genetically, we thought that we can maybe use these genetic properties that we already know very well to validate all the procedures that come before the linkage map building. So if I have good recombination fraction and maps like this, I can trust in all the other things that I did before. So the idea is to build linkage maps and to validate all the procedures. So when I say the procedures that come up before the upstream process, I'm talking since the DNA extraction until the genotype calling. And of course, here I do more with the bioinformatics tools, but with genetic maps, as the previous presenters also say, we can also detect like things that went wrong, for example, with contamination in the sample, because the pattern of the recombination in the map will not make sense. And to test it, we needed a way to easily go from the sequence to the linkage map. So we built these workflows, writing in workflow description language, also called as WIDO. And this language is the same that Broad Institute used to provide the best practice workflows. And is a kind of thing that we are trying to do here, like pick the best combination of software and parameter to build the linkage map, using the linkage map as result to check. And we built two of them. One is the empirical reads, that's for processing empirical data. And the other one is the simulated reads. And the empirical reads, you just go with your samples and the genome file, and you align the sequence to a reference using BWA, and then SNP calling with GAT key and freebase, and genotype calling with updog, polyred, supermassa. And by now, it's just for diploid species. I'm expanding to polyploid, and the only software that do the linkage mapping is OneMap. And if you use the simulated reads instead of going with your samples, you just go with information of the scenario that you want to simulate, and these two software will do the read simulation. This is the simplifier version of the workflow, but if you really look inside the workflow file, and I can generate these figures just with a tool and with the workflow file. This is a sub-workflow to simulate a single family. And then we generate these figures. The workflow are composed by tasks, that's the smaller units, and the tasks can be combined in sub-workflows that can be run independently. For example, I can just run Freebase if I want, and I can combine the subworkflows and workflows. And was what I did for the main workflows. And once the workflows is ready, I can use a single line to run all the things. And this is the single line. I use the Crowell. It's also a Broad Institute too, to do the management of the workflow. And once I do this, all the temporary files will be available. And it's only possible to run this with a single line, because each task is performed inside a container, a Docker or a Singularity container. So I don't need to install anything. It's all everything already available in these images. And Crowell also have compatibility with cloud and with high performance computing, including Slurm and other management system. And all the intermediary files will be available, but I did the workflows to output a compressed file. And this compressed file, a single file, can be uploaded in a Shiny app. And yes, I like Shiny apps. And I have like here a spot for the simulated, for the results for the simulated workflow. And this is for the empirical workflow. So you just upload here, and you can see and access several graphics of diagnostic of the maps built. The workflows just build maps for a single linkage group, because there are many combinations, like in total will be 34 combinations for empirical and 68 for simulated. But once you pick the combination of the pipeline that you want to proceed with, like the best combination, you can download the sequence and repeat the same procedure for the other groups. And I brought here just, I think I'm running without time, but I brought here two examples. This is rock curves. That's results from the simulated workflow. So here I call the SNPs with Freebase and JTKey, and I simulated reads with mean depth of 10 and 20. I take the read counts from two different sources, the alignment file and the VSEP file. And the rock curves here show the genotype probability reliability. And so if the genotype is wrong, I expected that the genotype probability to be lower. And if it is correct, I expected this to be higher. So curves that approximately, that is close to the left upper corner means that the software, the genotype software, can do this differentiation very well. And just a overview, like UpDog and PolyRed can perform really well in all scenarios simulated. And this is an example of the empirical reads output. And I performed in deployed roses, and it's just a subset of the chromosome 1. And this is the number of markers of the subset is 37 centimorgans, the subset. And this is the distance in centimorgan of adjacent markers, according with each one of the pipelines. And as conclusion, yes, we built this workflow to be able to select the best pipeline and parameters, and these parameters can be changed inside the workflow to be tested. And this structure, the combination of Widow, Docker, Crowell, and Shine makes the Reads2Map workflows a multi-platform you can run in your local computer. I would not do this with entire dataset, but you can do this locally or in HPC or in cloud environments. And this is practical and user-friendly and reproducible, mainly because of the containers. And if the upstream softwares do updates, what I know that everybody is doing updates, it's simple to change here because I just need to change the version of the software in the Docker images. And I'm adapting Reads2Map to, instead of using one map, use MapPoly and PolyMapR in the last test, and to be able to do this with polyploid species. But I will need to talk a lot with Marcelo and Peter, because I am the maintainer of OneMap too. So every change that I need to do, I did very, very easy. And this changes all the updates that I did to be able to OneMap be compatible with Reads2Map is already available on CRI. So I would like to thank you, all the collaborators. So from the University of São Paulo, from the colleagues here, the NC State University, the Federal University of Viçosa, the private company Mendelix, and the Roslin Institute, and the funding agencies and USDA. Okay, thank you.